Welcome back to the Deep Dive, where we turn stacks of dense research into, hopefully, immediate and impactful knowledge.
Hopefully.
Today, we are taking a massive shortcut right into the fundamental language of the human body. We're talking about cellular conversation.
That's right. We're diving into cell interactions, or CCI. This is the dynamic complex signaling network that dictates basically everything, from how your immune system responds to a threat to how a complex tissue develops and functions.
It's not just basic biology, then.
No, not at all. This is the core operating system of your physical existence.
I like the analogy of thinking about tissues, say your liver or your brain, not as just a static pile of cells, but more like a bustling city. The cells are the residents, right? And they're constantly communicating. Some messages are sent by direct physical contact, like a handshake.
Right, or passing a note.
Exactly. Others are paracrine, which is sort of like short-range radio waves across the street, or even autocrine, sending a note to yourself.
But the key, and this is absolutely central to the source material we're looking at today, is that these interactions are intensely dependent on distance.
Precisely. And that's where the problem comes in, because we have these incredible new technologies.
Oh, absolutely. Like imaging-based spatial transcriptomics, or ST. It literally gives you the GPS coordinates of every single cell and what genes it's expressing at that moment.
So we have the map, we have the resident list, but we can't translate the dialogue. What are the big hang-ups with the current methods?
Well, the sources outline 3 critical failures. First, our existing methods are way too reliant on incomplete databases of known ligand-receptor pairs.
Okay. What does that mean in practice?
It means that a lot of these cutting-edge imaging platforms, they only measure a limited panel of genes. So that crucial interaction data, that ligand or that receptor, it might be sparse or even completely missing from the data set.
So if the specific handshake isn't in the library, the method just assumes the cells aren't talking at all.
Exactly. It's a huge blind spot. The second failure is ignoring what we call cellular heterogeneity.
Meaning not all cells of the same type are the same.
Precisely. Traditional methods treat all cells of a type, say, all excitatory neurons in one layer of your cortex, as if they're functionally identical. But we know, biologically, that a cell's precise local environment radically shapes its state and its function. The old models they ignore this variation.
And the third failure?
It's poor spatial encoding. They just don't sufficiently incorporate the physical distance between cells into the calculation. They might see 2 cells are neighbors and assume it's a strong conversation.
When in reality, the signal might decay really quickly, or the receptor just isn't there to pick up the call.
You got it. It's a very naive assumption.
So science needed a shortcut, something that integrates all 3: location, gene expression, and that individuality.
And that's the solution we're dissecting today. A new model called GITIII, that's GITIII, the graph inductive bias transformer for intercellular interaction investigation.
That's a mouthful.
It is, but the concept is actually very elegant. GITIII uses a modified AI language model structure to infer these conversations at a truly granular single-cell level.
And it leans into that linguistic analogy, right? The idea of cells as words.
It does. It conceptualizes individual cells as words, and the surrounding cellular neighborhood, the microenvironment, becomes the context or the sentence that profoundly shapes what that central word means.
So the mission of this deep dive, then, is to see how this AI takes the location and the context, that surrounding sentence, and figures out precisely how much influence that sentence has on the single central word.
Okay, let's unpack that core idea, because the whole mechanism really hinges on a conceptual shift in what the model is even trying to predict. So before we get to the prediction, what are the raw ingredients? What does GITIII need from the data?
It starts with the foundational data you get from spatial transcriptomics, so a complete gene expression matrix.
Okay, so a list of all genes and their activity levels for every cell. Right. Then you need the precise spatial coordinates for every cell, and finally the cell type annotation, just simple labels like astrocyte or microglia.
Got it. Now, this next part, I think, is maybe the most revolutionary piece of the whole system, this idea of cell state expression. Can you break down how GITIII separates the signal from the noise here?
This is the crucial decomposition, you're right. GIII takes the entire gene expression profile of every single cell and breaks it down into 2 distinct components.
Okay.
First, you have the cell type expression. This is just the textbook definition. It's the average gene expression across all cells of the same type in the entire sample. You can think of it as the cell's baseline identity.
So that's the, let's say, 90% of the signal that just says, I am a macrophage.
Exactly. And the second part, which is the real target, is the cell state expression. This is defined as the deviation of that one specific cell's gene expression from its cell type average.
Ah, so it's the bit that's different.
It's the bit that's different. That deviation is the critical signal we're hunting because it's presumed to be driven by the local environment.
Wait, let me make sure I get this.
Yeah.
So if a cell is 90% defined by its inherent type, and maybe 10% is influenced by its environment,
Yeah.
GITIII literally ignores the 90% and only tries to predict the 10%.
Yes.
Why is that filtering so revolutionary? Aren't you throwing away useful data?
It's a brilliant trade-off. By isolating and focusing only on the deviation, we cut through all the noise of the core cellular identity. We stop trying to predict the known definition of a macrophage, which would be easy for an AI to do but would tell us nothing new.
I see.
Instead, we force it to focus entirely on predicting the functional consequence of the local neighborhood. We are quantifying the influence of the sentence, not just redefining the word.
That makes perfect sense. It forces the model to learn about external influence. So how does it gather that context, that sentence, for each cell?
For any given central cell, the word we're trying to predict, GITIII constructs a cellular neighborhood, which is just a subgraph, around it. It identifies the k nearest neighbors. Typically, the default is set to 50.
So it looks at the closest 50 cells.
Yep. That fixed size group becomes the input sentence.
And what's the actual machine doing the processing?
It uses a self-supervised single-layer graph transformer model, and the task is very, very focused. Predict the central cell's cell state expression, that deviation profile, solely from the information provided by its 50 neighbors.
You said single layer. When we talk about advanced AI, it's usually about deep, complex networks with many layers. Why stick to just one? Isn't that a limitation?
No, it's a very intentional design choice, and the reason is one crucial benefit: interpretability.
A black box versus a clear one.
Exactly. Complex, multi-layer graph neural networks can kind of obscure the path of influence. It's hard to trace back how a prediction was made. By using just a single layer, a modified block from a model called GITIII, the structure ensures that the model can directly calculate and quantify the specific contribution from each neighboring sender cell onto the central receiver cell.
So you know exactly who said what and how much it mattered.
It's designed to be a transparent conversation, not a black box prediction.
All right, a quick recap of what we've covered so far.
Good idea.
First, we have the core analogy.
Yeah.
GITIII treats cells like words, and their immediate neighborhoods like the sentences that give them meaning.
Right. And the main target isn't the cell's whole identity, but its cell state expression, that small deviation from the average, which is where the environmental influence lives.
And the engine driving this is a lean, interpretable, single-layer graph transformer. It's built for transparency.
And the goal isn't just to predict, but to quantify the specific influence of that neighborhood on the central cell's transcriptional state.
Let's dig into the mechanics now, specifically how GITIII prevents itself from cheating. How does it define the input features to make sure it's learning real signaling and not just, that similar cells hang out together?
This starts with the node feature, embedding how the cell itself is defined, and a really critical step called expression masking. This directly addresses the problem you just mentioned. Cells of the same type naturally cluster and have similar gene expression. Without a mask, the model would just learn that, astrocytes near other astrocytes are similar.
Which tells us nothing about communication.
Nothing. So the mask's rule is simple. If a neighboring cell is the same cell type as the central cell you're analyzing, its real observed gene expression is replaced by the average cell type expression we calculated earlier.
The baseline.
The baseline. Only if the neighboring cell is a different type is its actual observed gene expression used. This forces the model to look beyond simple proximity and shared identity. It has to find genuine molecular conversations that are driving that state change.
But isn't there a risk there? You're basically throwing out all same type or homotypic interactions. Aren't macrophages influenced by other macrophages? Don't you lose legitimate data?
That is the trade-off. You're absolutely right. But by focusing on the heterotypic, the different type influence, the model gets a much cleaner, more reliable inference of those external environmental drivers. The authors decided that masking was the most robust way to force the model to truly learn those intercellular patterns.
Okay, so that's the word. What about the sentence structure, the connection between them?
That's the edge feature embedding. This is where distance and ligand effects are encoded. The core of this is the ligand expression from the sender cell. But crucially, this expression value is explicitly scaled by a distance embedding.
And the sources mention that older models use the simple exponential decay for distance. Strong up close, drops off fast. But GITIII uses 5 different distance-based features. Why the extra complexity?
Because biology is rarely that simple. Different signals, say a growth factor versus an inflammatory cytokine, they can have very different distance-dependent patterns. Some might be short-range, some longer. By using 5 features instead of 1, GITIII can model the complex, nuanced reality of how interaction strength really varies over space.
And all this feeds into that specialized engine, the modified Turing encoder.
Yeah, we made 3 very targeted modifications to the standard graph transformer. First, we swapped out the original random walk encoding, which is better for sparse graphs like a social network, and we replaced it entirely with our more expressive distance embeddings. That's essential for the dense 2D layout of a tissue.
Makes sense. And the second change?
We built a distance scaler directly into the attention mechanism. This is a physics-informed step that ensures the attention the model pays to a neighbor naturally decays as the physical distance increases. It's basically baking biological reality right into the AI's math.
And the third was minor.
Very minor. We just removed the degree scaler, since every neighborhood has 50 neighbors by design. It wasn't needed.
So you have all this careful engineering, the input, the masking, the distance encoding, and the result is the final report card. They call it the influence tensor.
Yes. The influence tensor, which they call I, is the model's key output. And it's not just one score that says astrocyte talks to neuron. It's this huge interaction matrix that quantifies the precise impact of each neighboring cell on the expression of each specific gene in that central cell.
Whoa, so it's that granular neighbor A's effect on gene X, neighbor B's effect on gene Y.
All of it. A full breakdown.
Okay.
But before it's used, it gets refined. It's normalized into what's called a proportional influence tensor. And as a key robustness step, any interaction where the absolute proportional influence is below a tiny threshold 0.02 is just discarded.
So you're filtering out all the background noise and only looking at the strong, meaningful signals.
Exactly.
Okay, let's do another quick recap of those mechanics.
Sounds good.
First, expression masking is key. It prevents the model from cheating by forcing it to learn actual signaling between different cell types, not just similarity between the same cell types.
Yeah, right. Then there's distance encoding. It uses 5 features and a distance scaler built right into the attention mechanism to accurately model how biological signals really decay over space.
And the final output is the influence tensor, which is this incredibly detailed neighbor-by-neighbor, gene-by-gene report card showing exactly who is influencing whom and how. Now that we know how the engine works, let's look at the real-world utility. We'll start in the brain specifically, a mouse primary motor cortex dataset. Traditionally, methods would just see astrocytes as, well, astrocytes. What did GITIII uncover?
It achieved a level of resolution that's really unprecedented. It started defining cell subtypes based on their communication environment. Take those astrocytes. The original study found maybe 3 subgroups. GITIII identified 5 distinct subgroups, and they had this clear spatial separation that perfectly aligned with the known layers of the cortex.
Can you give us a specific example that ties a subgroup's function to its location?
Absolutely. The astrocytes in subgroup 1 were found strictly in layer 1 at the very top. But subgroup 5 was localized specifically in the white matter right underneath the cortex. And crucially, GITIII inferred that this subgroup was strongly influenced by nearby oligodendrocytes.
The cells that make myelin.
The very same. And these astrocytes showed increased expression of genes like GFAP and EGFBP5, which are known to be involved in myelination and regeneration. So it links their location, their neighbors, and their gene expression directly to a specialized repair function.
That is amazing.
Yeah.
It's extracting a functional role from the conversation itself. What about the neurons?
Well, the L23 IT neurons were previously a MES5 spatially indistinguishable subgroups. GITIII streamlined this to 3 subgroups, but these 3 had clear spatial separation. And we saw this incredibly powerful correlation. Interactions coming from L45 IT neurons were associated with the upregulated expression of a gene called ROAB in L23 IT subgroup 2.
Why does ROAB matter? What's the significance there?
ROAB is a critical marker, often associated with a neuron's layer identity and its connectivity patterns. The fact that the model explicitly linked its upregulation to the influence of layer 45 neurons, and that subgroup 2 was physically located closer to layer 4, it just shows the model correctly identified a highly specific, spatially constrained signaling pathway that dictates neuronal identity.
Okay, let's bring this out of mice and into human health. The Alzheimer's disease dataset from the CAID project.
Right. So the AD dataset first reinforced one of the model's core principles. Just being close isn't enough. We saw that even when cells were basically touching within 18 micrometers, the calculated interaction strength varied dramatically.
So you'd be shouting next to someone who's deaf, essentially.
A perfect analogy. Low ligand expression means a quiet conversation no matter how close you are.
And what did this tool uncover about the actual pathology of dementia?
This was the really striking part. When they analyzed 69 postmortem tissue slides, the model showed that patients who had been diagnosed with dementia had a statistically stronger overall CCI network strength between microglia, oligodendrocytes, and excitatory neurons.
Stronger, not weak.
Stronger, and specifically in the deeper cortical layers, it wasn't just that these cells were present or inflamed, it suggests there's a kind of hyperactive, maybe dysfunctional conversation happening between the brain's immune cells, the myelin-producing cells, and the primary signalers. And it's happening precisely in those deep layers, which correlates directly with cognitive decline.
So that shifts the focus from just general inflammation to a very targeted microenvironmental dysfunction.
Exactly.
Okay, that was a lot. Let's recap what we saw in the brain.
Go for it.
GITIII found hidden complexity. In astrocytes, it uncovered 5 specialized subgroups tied to specific cortical layers and functional roles, like white matter repair.
Right. And for neurons, it made that specific link between the ROB gene's upregulation in L23 neurons and the signals they were getting from their neighbors in layer 45.
And the big one for human health, the Alzheimer's insight. It identified a significantly stronger CCI network involving microglia and oligodendrocytes in the deeper cortical layers of dementia patients. This points to specific dysfunctional conversations as a potential driver of the disease.
Let's move from the brain to the tumor microenvironment, or TME. This is from a non-small cell lung cancer dataset. How does GITIII help us tell the good immune cells from the bad?
Well, in the TME, macrophages are these crucial players. They can be good guys fighting the tumor or bad guys helping it. GITIII identified a specific group, macrophage subgroup 3, which was heavily influenced by nearby tumor cells.
Okay.
This subgroup showed increased expression of a gene called SPP1. That specific molecular signature is a known red flag. It immediately identifies them as pro-tumorigenic tumor-associated macrophages, confirming their damaging role in helping the tumor grow. And what about the other side of that conversation? How are the tumor cells themselves being influenced?
We saw evidence of immune suppression. Tumor cells that were located near the boundary showed decreased expression of genes like GPNMB, EGFR, and KRT19.
And those genes are important for
tumor survival and proliferation. The takeaway is that the neighboring immune cells weren't just sitting there. They were actively sending signals to the tumor cells, essentially telling them, slow down, stop growing so aggressively, by dialing down their expression of these pro-survival genes.
And the gene ontology, or JAGO analysis, helped define the type of conversation.
Exactly. The JAGO analysis cleanly separated the roles. It showed fibroblasts were influencing tumor cells mostly through extracellular matrix remodeling. They're like the architects, physically changing the environment.
While the macrophages were different.
Completely different. They were modulating tumor cells through direct molecular control things related to cell cycle regulation and the p53 signaling pathway. Direct influence over life and death.
What really gives this model authority, though, is its robustness. Did the sources show that it works across different species, different platforms?
Absolutely, and that's a massive strength. We saw similar interaction patterns not just across species, the mouse brain versus the human brain, but also across totally different spatial transcriptomics technologies. The NSCLC data was from a platform called COSMX, while they also looked at breast cancer data from a Xenium platform. The core biological mechanisms seem to be conserved.
And finally, the big test. Benchmarking against all the other methods. How did it stack up?
In a head-to-head competition against 9 other tools, GITIII consistently achieved the highest intracell type variance explained by CCI. It did this across all 4 datasets.
And that metric is important because
it's the crucial one. It means its unique design, that focus on cell state expression, the masking, the distance encoding, was just far superior at finding the real biological signal that differentiates cells of the same type based on their local environment. The performance improvement ranged from 20% to over 200% better than the other methods.
So that specialized architecture wasn't just helpful, it was absolutely essential.
It was the key.
So let's wrap this up. The development of GITIII, it really feels like more than just a new algorithm. It's a new lens for viewing biology.
I think so. The key advantages are clear. It successfully estimates neighbor influence on specific genes, which oldie methods just couldn't do explicitly. And for future science, this is huge. It works efficiently even when ligand-receptor information is limited or sparse.
Which is a major win for all these new high-resolution imaging platforms.
A huge win. And of course, its explicit incorporation of distance gives it superior spatial encoding over anything that came before it.
But we have to be critical here. What are the limitations we need to keep in mind?
This is so important. The results from GITIII are magnificent for generating hypotheses, but they only indicate strong association, not direct causation. That incredible finding about the stronger microglia network in Alzheimer's. It needs rigorous experimental validation in a wet lab to confirm the mechanism.
Got it. Association is not causation.
And while the model cleverly bypasses the sparse ligand-receptor data problem, the consequence is that we still don't always know the full details of the downstream signaling pathways.
The functional consequences of the conversation.
Exactly. We have this precise map of who is talking and how loud they are, but not always the detailed molecular script of the reaction that follows. That's still the next frontier.
Which leaves us with a final provocative thought for everyone listening. If the health, the function, the state of every single cell in your body is measurably influenced by its closest 50 neighbors, and we can now map those cellular conversations with this level of resolution, what previously incurable disease might be solvable if we learn how to subtly shift just one cell's local sentence? What specific microenvironmental conversation would you investigate first to find a cure?
Until next time, keep digging deeper into the sources.